And that gets us to the liver. The liver is the largest exocrine gland in the human body, and it lies in the upper right corner of the abdomen, just below the diaphragm. Sorry about that. It secretes the bile, and the bile enables us to absorb fat. The bile acids in there actually do. The bilirubin gives it the green color. So if you actually, if you look at the cadaver and you see the viscera, you see this green thing, and it's very prominent in it, and that's the gallbladder, and the gallbladder stores the bile. And the green color is from the bilirubin, and that's the end product from hemoglobin breakdown. Other substances that we have in this bile are cholesterols and minerals, and they are excreted by the bile. They just get, then the bile can also be a means to get stuff out of the body that we don't want to have in the body. The liver metabolizes carbohydrates, proteins, and fats, and helps detoxify us by inactivating medications and poisons. It also synthesizes many blood components, albumin, clotting factors, and so forth. So that's a very important part the liver does. The liver, if you have a function that you don't understand who does it in the body, the liver is a very good target for that, because the liver does a lot of things. The liver is a gland, so it constantly reproduces, but the liver also does a lot of taxing things and gets beat up pretty well in our modern life, with all the alcohol, with all the toxins that come from, well, all over the place, including the air, the water, and so forth. And so here is a list of foods that help cleanse the liver. We have from grapefruit through beets, carrots, to different salads like arugula, for example, or spinach or avocado. So all that kind of green stuff, and of course turmeric. Turmeric is good for everything. Turmeric is pretty good because it is anti-inflammatory. When we look at the anatomy of the liver, we look from the front. The liver has a right lobe and a left lobe. Those are the big ones we could see. And they have a ligament that goes down. That's the falciform ligament. And it becomes the round ligament inferiorly down here. And in the old days, when you were not quite born yet, that was the umbilical vein. So that's part of that structure that is in the belly button. Facing the intestine, so facing backwards, the posterior end, the liver has what we call the liver gate. That's the porta hepatis, which is the entry and exit point of the blood and the lymph vessels, as well as the nerves in and out of the liver. So again, we want to have it be in one place, like in the lungs. We had the hilus, and there we have a quadrate lobe, as well as we have a caudate lobe in the back part. Quadrate lobe's down here, the caudate lobe above there. Microscopically, this is, if you touch this, it's sponge-like kind of connective tissue, and inside there's a lot of divisions, and we get these hexagonal lobules that are about 1 millimeter to 2 millimeter in diameter that go up and down. You can see them like here, the way they're structured. We got a central vein that drains the lobule in the middle. There it is, a central canal down here. And from the outside, we have a branch of portal vein and hepatic artery that comes in. We have the hepatic artery, we have a hepatic portal vein. And those feed each lobule. And then we have, so they go inward like this. They feed into the system this way. And then we have a biliary duct that goes the opposite way, that leaves it and then becomes a bile duct, and that's the bile creation situation there. And between those canals, we have hepatocytes, and they run tiny sinusoids in which the peripheral blood runs towards these central veins. So we get towards these central veins, and in between we have these are the sinusoids, and we have these hepatic cells in this area. And the hepatic cells do the work. They clean the blood. And then we have also Kupffer cells, and the Kupffer cells are right here. You can see a Kupffer cell right here. And the Kupffer cells are the immune cells. They're macrophages, essentially, that are residing. They're like the dust cells in the lung. They just do the function of macrophaging inside the liver. That's pretty much it. except then there at the bottom that the bile goes the opposite direction. All right. Let's see what's next. Oh, look at that. Of course, the bile is going to be made, and it's going to be having to go to the gallbladder where it's stored, and it is also concentrated right here. The gallbladder is in the back of the liver, and it holds about 30 to 35 milliliters of bile. And the reason, again, why we need the bile is because fat purely is soluble in water, so it clumps together. And we get the bile, which has a polar end and a non-polar end, and it could go in between those fat cells and disperses them. It emulsifies the fat into fat droplets. And so the non-polar end of these little molecules, the bile acids, go towards the inside and hold the fat, and the polar ends are happy in water, so we can have small droplets that then the system can absorb. They're small enough to absorb. Otherwise, the fat just gets, goes through the system. So when you have the gallbladder removed, you still have bile, but it's small little dripping into the system. It's not from the gallbladder. And the reason you need the gallbladder removed is because sometimes these crystals, they become crystals and then they get lodged in this bile duct, and that really hurts. So they have to remove it. Gallstones. All right, and that almost brings us to the end. So we're going to have an overview of the different digestions. So we have fat digestion. Most ingested fats are triglycerides, and they have to be split by lipases, which are enzymes, into fatty acids and glycerides. We have lingual lipase, which is in the tongue. That starts the chemical digestion process. So yeah, we have a little bit of digestion of the fat as well, not just the carbohydrates. But the carbohydrates, the classic. And that's about only doing 10 to 30% of the ingested fats, while the rest starts with the pancreatic lipase, and that's where it really takes place. The products from the tiny globules, which we can call them micelles, are passively diffused through the epithelial cells into the lymphatic vessels. And the lymphatic vessels deliver them to the bloodstream, from where they reach the body tissue. And that's what fat does. When we look at carbohydrates, depends on the chewing amount. The chewing is important in carbohydrates, because if we just swallow the thing whole, the amylase is not going to be working on the carbohydrate. But the chewing process and the saliva will digest the carbohydrates by salivary amylase, or start the process. And then pancreatic amylase further splits the carbohydrates into disaccharides, which then are split into monosaccharides by disaccharidases. See all these words? But they kind of make sense in some weird ways. And then they can be absorbed, and they have to be actively absorbed, and some get passively. And they go into the bloodstream and then reach the liver via the hepatic portal vein. If we have a deficiency of certain enzyme, particularly lactase, some of us cannot digest milk protein, milk carbs, which is lactose, milk sugar. And so therefore we retain those people retain a lot of water if they drink these products or eat these products. And because we have osmosis, we have larger molecules that are staying inside the system, and they absorb, they will pull water with them. And this causes a lot of gas formation and diarrhea. And so what we can do is we can eat lactase and help us digest the lactose that way. But that's what, when we say lactose intolerance, that's what that means. Okay, then we get to protein digestion. The first step in protein digestion takes place in the stomach, when the strong acid denatures the protein, makes it easier to process. So then pepsin splits that protein into the polypeptide chains. Talked about that already. Once it's into the water, we have pancreatic enzymes, and look at those names: trypsin, chymotrypsin, carboxypeptidase. Again, they all make sense. They're complicated names, So I'm not going to worry about memorizing them, but you will have a sense of what they mean when you see them. And what they do, all those do, they attack the peptide bonds. They go and chew up what's holding the amino acids together, and they make them into di and tripeptides, and then into amino acids, single amino acids. And those will be actively absorbed by the epithelial cells before we get into the bloodstream. About 10% of ingested protein will get all the way to the large intestine, where it will be broken down by bacteria. And so that's, again, another reason why we need the commensals there. All right, well, that was a good session. I hope you learned something. See you later. Take care.